All right. Well, hello everyone, and welcome back. We've got a very interesting show today. Instead of going after Frank Turek again, we're going to have a more positive show. We have a guest. We have. I should have asked. I should have asked beforehand. That was my fault. Um, how do you prefer that I? What do you prefer that I call you?
Yeah, the name is Inaki,
Inaki.
but don't worry, if you say Inaki, it also works.
Okay.
Yeah, don't worry, you can try it. It will be
Inaki.
fine.
Okay. No, that's no problem. I just realized I forgot to ask what you prefer to go by in the pre-show. Welcome to the show. Glad to have you.
Thanks a lot. Glad to be here.
Yeah, I've been looking forward to this for a while because I have read a number of your papers, and I saw you were on Twitter, and I was like, heck yeah, let's see if we can get him on the channel, talk about some stuff with him. Because I did actually get you've probably heard of William Ratcliffe. I had him on the show. It's been a few months, right, Peter? A few months since we had Ratcliffe on, something like that.
Great guy, I know him.
Yeah, that was a lot of fun. That was a really interesting conversation. If you guys aren't familiar with William Ratcliffe, look up his yeast experiments he's done.
Yeah.
So you also do a lot of stuff with like the evolution of multicellularity, especially as it pertains to like our close relatives, looking at like homologs of different genes in like our close protistan relatives, right? So
Yeah.
if you want, would you like to tell us about your background and some of the research that you've done, just in case anyone doesn't know?
Yeah, sure. I'm evolutionary biologist. I work in the Institute of Evolutionary Biology, actually, in Barcelona. And I have been interested in the evolution of life in general. So I have worked in the evolution of bilaterial animals, and the last 10, 15 years on my own lab, I'm mostly focusing on understanding how unicellular organisms became multicellular animals, so how this transition occurred. And to do that, we compare unicellular relative of animals, the closest unicellular relative of animals, with animals, and try to infer how was the unicellular ancestor.
Absolutely. I'm really glad that there are people out there studying like ichthyosporians and philisterians and things like that, because I'm not TAing botany this semester, but I still tutor students, and so of course we talk about different protists and algae and stuff like that. And I was describing the opisthocons to them, and I was like, well, basically, you have your three important groups. You have fungi, choanoflagellates, and animals. We're not really going to talk about all these other guys because, we really just tell them about the fungi and their ecology, and then choanoflagellates and how they're close to animals, and then we very briefly mention animals, but that's about it. And so we're like, sorry, if you want to know about these other guys, you'll have to go read the literature on them, because yeah.
Yeah, but you know how things work. Sometimes the ones that people know less are the ones that can be more informative. So
That's true.
yeah, you have an issue.
Yeah.
I thought relates are very informative as well. So in this case, all of the unicellular relatives are very informative.
That's good. Yeah, I mean, these are also they're predominantly sophomores, and we don't want to overwhelm them with with information. But yeah, so the with the evolution of multicellularity in eukaryotes, how many times approximately would you say that transition has been bridged?
Yes, several times it has occurred independently more than 20 times
Mm.
in the evolution of eukaryotes. Some of them are more complex and more famous, and others are less known, but at least more than 20 times.
Mm. So does that speak then to the relative ease at which multicellularity can arise from unicellularity? Do you think?
Yeah, I think so. I think it talks to two different things. So first, it may not as difficult to obtain,
Mm.
being multicellular, and the second thing is probably advantageous. There is some advantage
Mm.
of being multicellular. So otherwise, I mean, so the important point also is it's a major evolutionary transition, but it's the only major evolutionary transition that has occurred more than one time. All the other major evolutionary transitions, such as the origin of life, or the origin of a prokaryotic cell, or the origin of
Mm.
humans, it has occurred only once. But multicellularity has been acquired several
Right.
times. So not so difficult to obtain, and at the same time, it has to be advantageous.
Right. Yeah, absolutely. Um, yeah, and it's been achieved. Like there are some groups who just do it. They're almost indecently eager to evolve multicellularity, like the various chlorophytes and things. You just look at them, and they're all doing multicellularity in different ways. They've independently reinvented multicellularity like dozens of times. Like the vulvacines and all those guys. But so uh, would you say there's really a difference between like simple and complex multicellularity, or would you say it's more of a gradient? How do you typically think about that?
Yeah, yeah, I would say it's a gradient, like most things in biology. But in order to differentiate, we tend to differentiate between simple and complex. There have been some authors that define complex as having different cell types.
Hmm.
Some authors can even go farther and say have different cell types, they have an embryonic development. At the end is how you use these words. In my opinion, there is a
Hmm.
gradient, and also sometimes whenever we think it's simple multicellularity, you can be surprised later on when you analyze better, and it can be not as simple as you originally thought.
Right. Right.
But yes, for sure, there are organisms that are much more complex because they have different cell types and they have more complex development. And that will be, for example, plants and fungi and animals and red algae, for example. Those will be probably the ones that are more complex.
Right. Yeah. Everything in biology is more complex than it seems at first.
Yeah.
With the Ratcliffe experiments, the thing that he found was predominantly what causes multicellularity, at least at first, was in the to generate the snowflake phenotypes was or well, no, he didn't figure it out, but they used it in a later paper in their later work. It was like Ood et al. 2013, I think, which did the genetic testing. But they found it was like a single gene involved in septum dissolution, which basically it broke, and so it wouldn't dissolve the septum. So cytokinesis would happen. So the actual so for anyone who's not aware, the cytoplasm of the cells, all their organelles and stuff would split, but the cells wouldn't separate like they normally would because the septum, which is this little wall that forms during that process, would not dissolve, wouldn't dissociate, and so they couldn't uh, butt off from each other. But which is also interesting because multicellularity requires clonal reproduction, right? Of cells, right? It's not coloniality. It's not a bunch of cells that are genetically disparate aggregating together. It's always like the clonal reproduction of cells, right?
Well, I mean, animal multicellularity is clonal now, for sure.
Right.
But there are organisms like Dictyostelium, for example, that is aggregative multicellularity.
Right.
So I think you can have both options, but the ones that are more complex now, they are clonal. Which,
Right.
by the way, that they are clonal, it doesn't mean that they originated by clonal division.
Mm.
They could have originated by other means and then develop into clonal. At the end, all eukaryotes have clonal division. So this is like, this is for sure.
Right.
You are clonal. How do you get to the first multicellular organism? That's another story, and I think we still need more data.
Yeah, absolutely. Also, hello everyone who's in the chat. Welcome. Yeah, you did I believe you did a paper where you looked at genes that were involved in the transition to multicellularity in animals, and one of the things you found was that like early animals must have undergone lots of gene duplications, right? Wasn't that there was like a bunch of gene duplications? Was it? What is the? I don't quite remember. What was the reason for that sort of increase in gene duplication events?
Yeah. So actually, what we see is that there is a general tendency of increasing genes from far away from the opistocon ancestors. So in the lineage leading to animals, you see a continuous increase of genes and genes that are important for multicellularity. So cell addition genes, transcriptional regulation, cell signaling. This is continuous. So what happens also is that at the origin of animals, there is even a wider increase and duplication, for example, of ancestral gene families, which it doesn't stop there. So it continues. And then when you get to eumetazoans, it continues and you have more. And when you get to bilaterians, you have even more gene duplication. And when you get to vertebrates, you have even more gene duplication. This is a continuous. So I think some gene families are clearly very important for animals, and because animals become with they have more tissues and more cell types, then they use different genes for different more specific, more fine-tuning
Right.
things. That's why there is a continuous explosion or division of ancestral gene families, duplication. But we also observe that there is gene loss also at the origin of animals, specifically metabolic genes, and there is also fusion of genes. So there is several mechanisms that are working that, and one of the mechanisms that it's also clear that is very important is co-option, so recycling ancestral genes that will work into new functions. So I think it's a combination of everything: new genes that are completely novel, new genes that come from ancestral protein domains or fusion of different genes, and then co-option recycling ancestral genes.
Absolutely. That and those processes, especially co-option, that's something that I try in videos to communicate the importance of recycling genes, because there are I live in the US, so there are some pretty bad misconceptions in the general public about relating to evolution. One of them is that for new functions or new proteins, etc., you have to have new genes, and that just simply isn't the case for most things. It's about co-opting, reusing what you already have, or in some cases duplicating and then neo-functionalizing one of the copies, or something like that. And that's absolutely fascinating, especially in the case of one of my favorite examples of that is there's an ice fish. So there's this cod that lives in the Arctic in the Antarctic, and it has like 40 duplications of this partial gene. It's just a couple of amino acids that got duplicated over and over and over again, and they use it to prevent ice formation in their bloodstream. What the original protein, the full thing, was a trypsinogen, so it was used in breaking down trypsin, but now it's clearly no longer using that. It's just this one little bit that was useful just kept getting duplicated. And I think that sort of stuff is absolutely fascinating. I'd love to do research like that in the future. I think that's all very interesting. And that also ties in with the origin of animals, because his work is like your work and Nicole King and all these other researchers have shown our close relatives, our close protistan relatives who are unicellular, possess a lot of genes that we also have and that we've duplicated for various functions, right?
Yeah, yeah. So I think actually many more genes than we were expecting when we were, studying or when we were starting all these projects. We thought, because you had the mentality that proteins are quite simple
Hmm.
and that animals are very complex. So I mean, we could never imagine that we will find so many genes, not only because they are shared genes, but also because there are genes that are extremely important for animal development, for example, or for animal multicellularity.
Mm.
And you could never imagine to have those genes. I mean, for example, you can find genes that are involved in organ growth. How can you find genes that are involved in organ growth in a unicellular organism, in an amoeba?
Right.
That we couldn't imagine that, or we can find genes like mercury, which is involved in gastrulation, which is one of the big moments of animal development, of embryonic development.
Hmm.
Uh, so why a gene that is involved in gastrulation, it's already present on unicellular ancestor, and it's now present in extant unicellular organisms that are not gastrulated,
Hmm.
not performing embryonic development. So that tells you a lot about the raw material that the ancestor had in order to construct the new body plans of animals, and to create functions, and to actually recycle ancestral functions into new functions. But
Absolutely.
yes, I think one of the most important findings that we have had in the last years, in the labs, not only my lab, but the lab of Nicotine, for example, which is the leader on this field, is how many genes we have identified on those organisms, and
Mm.
how that this changed our perception of how the unicellular ancestor was.
Absolutely. Has there been a lot of progress on determining what these genes are doing in, or what the homologs are doing in this
Yeah,
way?
a little bit. So this of course it's another step, because then you need genetic tools in some of these organisms to understand the function and so on.
Mm.
So we have been working very hard in order to get genetic tools. So for example, now we have in our lab we have genetic tools on 3 different organisms that are close related to animals. So this can tell you much more. And we know, for example, that mercury, for example, which is a gene involved in gastrulation, it seems to be clearly involved in cell motility, which at the end is more or less what gastrulation is about. It's about cell motility. So it's not only that the gene is conserved, but seems to be conserved. At least part of the gene regulatory network is
Mm,
conserved,
okay.
which is quite something. If you think how many years of evolution
Yeah,
we have
right,
had.
right, absolutely. And they have, much shorter generation times than we do, and so they can accumulate mutations
Yeah,
differently from us and all that sort of stuff. Yeah, absolutely. But yeah, that is fascinating. I remember reading it was one of Nicole King's papers from the early 20 teens, where she was describing all these different oh goodness like tyrosine kinases and all sorts of stuff that the choanoflagellates have, and it's like, huh, wonder what they're using these cell signaling enzymes for, that we use to, you know for talking between our cells, which is, yeah. And they've
yeah,
even identified like a hedgehog homolog, I think, at this point, haven't they?
yeah. It's a
Okay.
potential homolog. I don't think it's a clear
Okay.
homolog, because it's only one part, but it will be like the precursor of
Right.
Pitchfork. Yes. So which is also present in other organisms. And protein tyrosine kinases, they are present in many of those
Right.
organisms. Interestingly, the receptor protein tyrosine kinases are much more conserved with animals, and the cytosolic protein tyrosine kinases are less conserved. So it's a little bit of
Mm.
distinction between one of another. But there was even a further increase of protein tyrosine kinases and phosphatases at the origin of animals. So that was clearly something very important for the ancestor that became even more important in extant animals.
Okay, awesome.
And, and then besides the genes, so it's also the mechanisms of gene regulation and genomic regulation. So because you could say, well, the genes are there, but what about the mechanisms of gene regulation and genomic regulation? And one of this another important findings is that we also only analyze one of these organisms, but we find that it shares many of the mechanisms of genomic regulation and gene regulation with animals. It actually has all the same, more or less, mechanisms of cell type differentiation as animals has. So you could say, animals, in order to differentiate into tissues, they use alternative splicing, differential alternative splicing, differential phosphorylation, differential non-coding RNAs. So all of those things are being used also on those proteins.
Yeah, absolutely. That is really cool. That's really neat. Yeah. So in terms of this process, this the sort of evolution of multicellularity in our lineage, when approximately do you think that the first animals really evolved? Do you guys have like a time on when you think that process itself occurred?
Yeah, I mean, we follow what other authors are finding on that, and the fossil record will say more than 700 million years ago, and the molecular clock will say even farther than that,
Right.
900 million years. It's probably somewhere between those ages. Precambrian probably, which everything was more or less static. Yeah, I guess. I mean, I'm just following what the experts on that are
Gotcha. Yeah, I've seen
telling us.
stuff like that. The molecular clocks tend to point to like very ancient dates. So not quite as ancient as they used to, because there used to be, I saw ones that were like the origin of animals is 1 billion years ago,
Yeah,
and now it's closer to like 800 million years ago. But still, yeah, there's a, and the
yeah,
paleontologists, at least the ones I tend to read, seem to argue more for like 650 million years or something like that, because and that was actually, I guess
yeah,
that sort of segues us into the sponge thing. There's the earliest evidence of animals right now is these sponge sterain biomarkers from Oman, right? And
yeah.
there's been a little bit of controversy over that, to put it mildly.
Yeah.
So where, well, I mean, I know where, based on your papers, but for the audience, where exactly do you fall on that controversy?
Yeah, I mean, I've been hesitating a lot, but what I can say is I'm not sure that the biomarker is specific to sponges. We should work. The thing is, it's used as a specific to sponges, but the thing is, we don't know whether this is present in other
Sure,
organisms.
sure.
And some of the preliminary data shows that potentially some of the organisms that we work in the lab may have a very similar biomarker. So, I don't think you can use it as a biomarker unless we have a better understanding of the lipids on different eukaryotes,
Right.
because we may be confusing. We may be thinking it's specific to sponges and it is not.
Right.
And I wouldn't be surprised, because I mean, I've been surprised so many times in biology that I wouldn't be surprised if that's not a specific biomarker anymore.
Sure.
So I would take it with, I will be cautious about saying that.
Right, exactly. And that's, um, yeah, I've kind of, I kind of share I share that sentiment, um, because the argument, or well, the conclusion at this point is sort of, we can't say, it's not sponges, but we also can't not say it's some extinct group of algae also, because that was one of the things from the outset. The first paper by, was it Gold? Was it David
Yeah,
Gold?
yeah,
And they were arguing it's similar. It's a similar composition to pelagophyte algae, but it's slightly different, which argues more for sponges. And then someone else, another team came along and was like, well, it's also sort of similar to these these lipids that some rhizarians make, and but it's a
Yeah,
little bit slightly different from that. So it's
yeah,
yeah, yeah. So there's that
yeah.
whole
We need more data to have a very clear definition whether it's specific to sponges or not.
right.
In any case, I don't think this will change the age because it's probably
Hmm.
sometime on these
Right.
650 million years. So
Right, and the and that's also really a there's been a revolution within like late Precambrian um fossils within the last 20 or so years, because if you look at popular level books like Stephen Jay Gould's Wonderful Life, and the Precambrian fossils were more or less written off, like they were strange, we don't really know what they are, so we're just going to kind of set them aside, and then the Cambrian explosion happens. But now we're or the paleontologists are seeing more and more that a lot of these are actually animals. They're not lichens or fungi or whatever else. Are actually early animals, and so this is providing this sort of maybe not long fuse, but an intermediate fuse for the Cambrian explosion. 30 or so million years later.
yeah, yeah. I I think it makes sense actually to think that animal-like or multicellular-like animal-like organisms were appearing before,
Mm.
and you know we do have the Cambrian explosion, but I don't. I mean, we still it's not clear whether it's an explosion of in the fossil record, but it's a real
Right,
explosion. I think both options are still open.
right.
Um, but sometimes you need. I mean, usually you need a long evolutionary history path in order to get into something
Mm.
all the diversity of animal body plans. So you know, uh, and that's why when you check molecular clocks, I mean, and there is a difference with the fossil record.
Mm.
Right? It's because molecular clocks are checking directly the gene, so which is probably is a leading indicator. It's telling you something that later on will appear in the fossil
Right.
record. So
Absolutely.
yeah,
Yeah, that's true. And I mean, the fossil record is, as patchy as it is, you can only get, only so much resolution with certain events in evolutionary history, and so yeah, you're right. I agree. I tend to fall more between the long and intermediate fuse hypotheses that rather than like the short fuse, which was popular with paleontologists until the past like few decades. But that could just be me.
yeah,
um, which also sort of uh, segues us into some of the research that you have done. You mentioned body plans, and so you've also done a lot of stuff with like more basally derived animals, animal phylogeny. And so, out of sheer curiosity, I guess, where do you fall on the Porifera versus Ctenophora sister hypotheses?
uh, I will fall in the place in which it's still unclear.
Oh,
That will
no
be
fun.
but I wouldn't disregard the Tino for first hypothesis. I think the data it's also quite strong,
Hmm.
and and sponges I mean could perfectly be secondary derived. So
Sure.
I don't see any problem on that. But I'm not 100% convinced in neither of the two hypotheses.
Fair enough.
Um, why actually there has been so much debate on that I'm now collaborating with some people here in Barcelona to do our own analysis, and also with people in UK to do our own analysis now and try to see where our own analysis brings that. I have to say that some of the the trees that we were doing for the origin of animals, in some of the trees we got ctenophores
Hmm.
first, and some of the trees we got sponges first. So uh, we want to know. Actually, I really want
Yeah.
to know. So let's see. Let's see if we will know something more in the near future.
You'll have the final say on it. This will be it.
I don't think I will have the final set, but I would say another. I will have another
Right.
set. At least, at least I want to be more convinced.
Sure.
So if I'm the one involved in that and I can see what people are doing very closely, maybe I will be more convinced, because now I'm very open. I see very good researchers with very good analysis saying sponges, and very good researchers with very good analysis
Right.
saying ctenophores. So I don't know.
Right. I think there was a a King paper that said we should embrace the uncertainty in these phylogenies, just simply because of the time that has elapsed since these events took place, and so and all that sort of stuff.
Yeah, and I think that's very important to embrace the uncertainty. But at the same time, I don't want to embrace this uncertainty
Absolutely,
forever,
sure, absolutely.
because I think there are potential ways to solve that. Actually, the lineage leading to ctenophores is extremely
Mm,
large. So who knows? There may be a ways to break that line so that it's
right,
better, have a better phylogeny. So I would try to fight about embracing that forever. So for now, I totally embrace that it's uncertain.
right. Yeah, absolutely, and that is we've actually, so I'm doing a series. I don't know if you ever read the book The Ancestor's Tale by Richard Dawkins.
Yeah, sure.
So
Yeah,
we're doing a series on this channel where we are going through the tales, one by one, and because this book was written a few years ago now, we're sort of updating and expanding them. We're looking at like what did he say versus what does the data now say? Have there been developments? Is it the same?
yeah,
That sort of stuff.
yeah.
And so we're doing that. And one of the things we keep running into is well, one is long branch attraction, as you mentioned.
Exactly.
You have these long branches, and it's very difficult to uh, break them up. Or the converse of that, which is when you have rapid radiations, so you get a whole bunch of really short branches, and that's also problematic.
Yeah, yeah, yeah.
Why can't nature just make things easy for us, right?
It wouldn't be so much fun then.
That's true. It wouldn't be as fun. Um, I the one of the botany professors I've worked for, he's a fan of the neophyte hypothesis as being like sister to angiosperms rather than nesting within the gymnosperms, because there's a long branch. Neophytes have this really long branch and very few extant members, and so he has argued that that's that they only nest with gymnosperms because of this long branch attraction. Which, yeah, he may be right. Um, I don't have enough of an informed opinion to say one way or the other, but yeah, absolutely. Uh, do you is anyone in your lab working on like placozoans?
No, not now. We are not working with placozoans, although they are also very interesting,
Mm-hmm.
but we are not working with them.
I think it's interesting that as of right now, there's only one accepted species, which is a Trichoplex herons. But I've seen the literature on looking at the different lineages, and the researchers have pointed out, hey, there's enough genetic diversity here to warrant multiple species. They just haven't
Yeah,
been named really. They're just like strain such and
it,
such,
yeah, yeah,
which is another.
yeah.
Oh, sorry, go
And
ahead.
that's very important to realize. So most of the things we know. I mean, we know quite a lot about
Mm,
animals, but about some of the early branching animals, there is still a lot of diversity to sample and to do genomes and to analyze their genome composition and so on. So we still there is a lot of still to work, a lot of work to do that it may help um, on these tales, evolutionary tales.
okay. Um. Also, welcome everyone who's just now joining us. Yeah, you're right. There needs to be a lot more research out there. I don't understand why people don't think placozoans are sexy. They're absolutely sexy. I mean, you're
Yeah, they are.
um but also, and this is something we talked about before we came on. You were part of the group that helped break up the platyhelminths, the flatworms, um pointing out that one group, or a couple of groups, I guess, are
Yeah,
not actually um they're not platyhelminths, they are their own monophyletic group, separate
Yeah.
from the platyhelminths, and those are now go under the name Xenocoelomorpha.
Yeah.
What got you on that set of research?
Yeah, that was another important transition, which is the origin of bilaterian animals. So you transition from radially symmetrical animals to bilateral symmetrical animals. And bilateral symmetrical animals are, the fact of being bilateral, it also allows you to have anteroposterior and anteroposterior and ventral dorsal ACE, and then you can have a neuro system that goes into the anterior part, and then you got more complex body plan. So it's an important evolutionary transition as well. And actually, years ago, platyhelminths were considered one of the lineages that probably could be one, so the ancestral one to the rest of bilaterian. So we were actually trying to figure out whether that was true, whether platyhelminths were the sister group of the rest of bilaterial. And then we realized platyhelminths as they were defined were wrong. So there were two groups of platyhelminths. So there were acyls and nemertodermatids, and then the rest of platyhelminths. And the rest of platyhelminths will be inside the bilaterians, inside the Lophotrochozoa. And then acyl and nemertodermatids in our trees will appear in as the sister group to the rest of bilaterians. So there was some truth on considering platyhelminths like the first brand, but it was only a small part of the platyhelminth. So most of the flower mold will be in another part. And then later on, Xenoturbella, which is another kind of worm, was observed to be also closely related to acyls and nemertodermatids, and then the Xenocoelomorpha clade will be created. But the idea was to understand better, the evolutionary history of animals in the transition from radially symmetrical to bilaterally symmetrical animals. This is important because depending, there were ideas that the ancestor of bilaterians could be already very complex, but if you have acyls are quite simple, so acyls that don't have gut, and the nervous system is still very simple. So this will tell us that the transition to bilaterian was more gradual than was previously thought, So it's important to know which is the first branch, which is the sister group to the rest of bilaterian, as important as which is the first animal, which is the sister group to the rest of animals. So,
right.
Yeah, it's an important part of the tree of life.
Right. And so, how do you feel about the what is it, the the planula hypothesis, where there was maybe this neotenic event, so maybe the ancestor was something similar to the planula of cnidarians, which was ancestral to bilaterians?
Yeah. I should. I mean, I cannot answer now because I should revisit, because I was like a couple of years ago, I was revising that, and I got confused because I'm not sure if that theory was trying to explain the origin of animals or was trying to explain the origin of bilaterians.
Yeah.
And some of the assumptions, I think, were not correct under the new tree of life. So, but I cannot answer now, Jackson. I'm sorry, I don't remember the details. So.
That's no problem. But also, in sort of referencing what we talked about earlier, a lot of the genes that are involved in these acyls, like they're not coming up with whole new systems, or whole new systems aren't being invented like after the acyls branch off, right? It's a lot of duplication and co-option of existing genetic systems for new functions, right?
Yeah. Again and again, you see that in many parts of the tree of life. Co-option, very very important. Of course, there is duplication, but recycling ancestral genes, it's very important. Actually, every time so that happens also with the origin. So when early animal evolution, so when people sequence the first genome of sponges or cnidarians, the surprise was, wow, they share a lot of things with bilaterians,
right?
And that was like a big surprise. And then later, when we sequence conophyllosaureids and phylasterians and ichthyosauриans, wow, they share a lot of things with animals. So there are things, that they are much more ancestral than we previously thought. But also, I think a lot of that has to be with our own perception. Whenever we see a that we believe it's simple, then we are already supposing that that animal will have less genes and
Right.
less complexity. And so it's a lot about our anthropocentrism that we believe, there is a lineage acquisition and we are extremely complex here,
Right.
We are complex, but some of those organisms are also very complex and they work very well on their environment and they have a lot of genes and they function in very complex ways, so yeah.
Yeah, I absolutely agree with you. The anthropocentrism, it's a very good way to put it. I do also, well, okay, um so yeah, I did also want to ask I about going back to conoflagellates. And we kind of worked our way up the tree, but there was something I'd forgotten to ask earlier. And this is my own curiosity because I haven't seen really anything in the literature on it yet. I want to know if maybe you're working on it or you know anyone who's working on it. Um, the life cycle switch. So choanoflagellates have a largely haploid life cycle, and of course animals have a largely diploid life cycle, with the haplontic for choanoflagellates and then diplontic for animals. Do you know anyone who is looking at how that transition was bridged? Because I haven't seen really much literature on that yet. Or do you happen to know?
Well, we have a problem with that is that for most of the organisms, except actually for one guanoflagellate, we don't even know if they are haploid or diploid, and whether they
Okay.
do sex or they do meiosis or not.
Okay.
So until we don't have a better understanding of whether those organisms are haploid or diploid or whether they have different cell types and sex types and so on, it's very difficult to really tackle the transition because you need, remember, something very important is we are studying organisms that are extant now.
Right.
So they have evolved more than 600 million years of evolution since their ancestors.
Right.
So if you take one organism, you cannot infer that the ancestor was like that. So the only way to have a better idea of the ancestor is to have a good representation of organisms. And then if something is shared in several ones, then okay, then it could be in the ancestor. But otherwise, it's very difficult. So we are still lacking a lot of information of that. And as I said, there is only one guanoflagellate, and we know much more about this.
Right.
So until more, it's very difficult to tackle. And in our lab, we want to know more about these things, but it's not easy.
Absolutely. Yeah, I'm sad. I think choanoflagellates are absolutely fascinating organisms that just, I think it was 2 or 3 years ago, there was a paper that came out about how when they form colonies, they can actually move as a unit and they react to light, and they'll sort of bend like a tissue or something like that, which is super interesting. And so, yeah, it's very sad that there aren't lots of labs, out there all studying choanoflagellates because they're just so cool. Let's see. Do we have any questions from the audience? I see there are a few people in here. Maybe. So do you have do you have future directions that you want to go, or anything you want to share?
Yeah, I mean, future direction, I think, one of the, so we have sequenced a lot of genomes. We understand much better how the unicellular ancestor was. We even know that, as I said, genomic regulation, there was a lot of things shared with extant animals. I think now the questions are, how did actually cell type differentiation, animal cell types originated? And this is something that we are working with. Did spatial cell differentiation was already present before the origin of animals or not? This is something that we are also working with. How recycling of ancestral genes occur, the origin of animal embryology, how all of these morphogenetic processes that we see, such as the one that you mentioned of guanoflagellate, and we also have amazing morphogenetic processes in Ictiosporeans or in Phylasteria?
Mm.
Are they the precursors of processes that we are working on animal embryology or not? So this is something that we also want to analyze. And it's also very important to compare animal multicellularity with multicellularity in
Right.
other parts of the tree of life of eukaryotes, and see whether there is something that is shared, common or not, and whether we can create a common framework about the unicellular multicellular transition, so they are completely independent or not. This is something that I think we should also
Yeah.
analyze in the future. And I think it's also important to, at some point, maybe to, because multicellularity is extremely related to cancer, and some of the genes involved in the original multicellularity are related to cancer. I think it may be worth also analyzing, or even to have a theoretical evolutionary framework to understand cancer from the point of view of the original multicellularity.
Yeah.
So just to mention some of the things that it may be worth studying in the future, and of course having a better understanding of the diversity that is out there and that can inform us about the origin of animals.
Yeah. I do love that there are medical benefits to this sort of research, because I think a lot of lay people would sort of turn their nose up at it, like, oh, you're studying protists. Like, well, yeah, but they have a lot in common with us, and they can potentially tell us about things that go wrong in us, and so we can use our knowledge of them to
Yeah.
generate treatments ultimately, and that sort of stuff. And that's absolutely fascinating. Another thing, yeah, with the diversity, um, there was just recent, I think it was a year or two ago, there was a paper on Elizabeth Hehenberger.
Yeah. Yeah.
She found or named a clade, which is a pluriformia. So yeah, a new clade
Yeah.
of protists closely, or in our opisthocont group, so relatively closely related to animals. So yeah, we're still just finding stuff
Yeah.
everywhere.
Well, I don't think it's a new clade. The name is new, but it's actually a sister group to some organisms that are called Coraltricium dimatisporium that we already work on the lab.
Okay.
But they prefer to use a new name because now, instead of having one species, one taxon, when they will have two taxons. But certainly new species, we know for sure because we have been analyzing data, environmental data, DNA, environmental DNA, and we know for sure there is a lot of species and also new lineages that are, hiding from us in the oceans and in the rivers, and it may be worth checking them.
Absolutely. Yeah, the one of my favorite quotes is regarding the discovery of monoplacophorans in the deep sea, and it was we can now build a phylogeny of the whole of mollusca, when they were discovered, because they were thought to be extinct for like 400 million years or something like that. So yeah, absolutely. um okay, well, that sounds like a lot of really interesting research. I do look forward to reading a lot of that. Oh yeah, you mentioned with the ichthyosporians the morphology, because they switch right between like the different morphologies. There's one that has like one type of movement, and then there's another morphology that can move in a different way. All right.
Yeah, yeah. Actually, we have one organism in the lab that can have like 10 or 15 different morphologies, and it can change so quickly, and some of them look like worms, and then they can fusion, and then they can have a multinucleated structure. They can have like IFA-like stages. It's crazy, they can do extremely plastic organisms.
That's awesome.
And they can adapt very fast to the environment.
That's awesome. I love that. That's really cool. Well, don't want to take up too much of your time. So is there anything else you want to add? Anything you want to plug or advertise?
No, just if you want to follow our research, you can check on the webpage, which is multicellgenome.com, and happy to answer if somebody has any question. You can email or contact us, or if you come in Barcelona, just visit us if you want. And thanks for the conversation.
Absolutely. Thank you for coming on today. It was a pleasure. Honored to have you on. Um,
Thanks a lot, Jackson.
and thank you everybody who's in the side chat for watching. And we will see you all next time, next week.